So in this section, we're going to look into issues pertaining to the lipid itself in terms of how it can travel in your body, how can it be digested, and then later we look into how the cell membrane is built by the contribution of lipid or certain lipids. And then we look at the cell membrane itself in terms of its structure and also the function of cell membrane. In this slide, we're going to look into the challenges of lipid in terms of digestion as well as transport. Digestion concern, it needs to meet the enzyme, that is lipase, and only then say triacylglyceride can be acted upon by lipase to break it down to 3 fatty acid and glycerol. But the problem is the triglyceride does not mix with water environment. So how does it mix with the lipase enzyme? To solve this, there is the need of bile acid to help in emulsifying. That one we're going to learn in a while. Then you have transportation. Again, because triacylglyceride cannot mix with the water environment, the blood there is also water environment, the digestive tract also water environment, inside the cell also water environment. So how does it get it from fat cell to the muscle cell? In order to do that, you need a specialized transporter, and this specialized transporter is the lipoprotein that able to gather the lipid components and bring it to the cell. So this is the bile acid or bile salt, and you can see it has somewhat similar to the cholesterol structure because it is actually made with the cholesterol as the precursor in its pathway. And once it's been made, this is amphiperdic compound, and it can then be stored in the gallbladder, and when required it can be secreted into the digestive tract, and that's how it helps in the emulsification of lipid, and we will see how it will contribute in the next slide. So once the bile acid meets lipid big drops, what it does is, because of the empathetic nature of bile acid, it will make the big drops to become droplets and then even tinier droplets, and this increase in surface area will facilitate the lipase to act upon these droplets and then convert it from triglyceride to 3 fatty acid and 1 glycerol molecule itself. So next, to look into the challenge of transportation. If it's a short hydrocarbon chain of fatty acid, no problem. It can just travel through the blood by using the albumin that is found in the blood. Albumin is the most abundant protein in blood, therefore it can just bind to it. But when it's longer fatty acid, like 18 carbon, then it needs to be looked into a specialized transporter. Also, if it is a triacylglyceride or it's a cholesterol that is linked to a fatty acid, what they call it as cholesterol ester, all this requires this specialized transporter, here shown the LDL or low density lipoprotein. It's not that the lipoprotein carries the component of lipid, but it builds up around it. So in the core of the LDL, you can see it is made up of very non-soluble lipid components such as cholesterol ester. This is cholesterol plus fatty acid. Also TAG, triglyceride. And then those which are amphiperdic will be on the outer surface. So you have here the phospholipids, You also have the cholesterol, where the OH will be on the outer section facing. So all the things which are soluble, the amphipathic one, the head, the OH, all will be in the outer section, therefore easily can be soluble or can interact with water environment found in the blood, for instance. You also have this apoprotein. This apoprotein is the largest, in terms of size, largest protein in our body, and it is like a anchor or recognition part of the lipoprotein. When it travels through the blood, when then it comes to the cell, if there is a lipase there, it will recognize this apoprotein, and then it will cleave the triacylglycerol to become fatty acid. So that's the purpose of the apoprotein. So these lipoprotein are divided into a few types, and here shown the types of lipoprotein. You have the chylomicron, is the largest in terms of size compared to the other lipoproteins. This chylomicron will originate from the digestive tract. So all those diet fat or lipid will be taken up by chylomicron, and then it will travel to the cells and then come back to the liver. So that's the job of chylomicron. Then you have VLDL, or very low density lipoprotein. This one originates from the liver. So liver actually makes fatty acid, triacylglyceride, even makes cholesterol. All these will come into the VLDL, and then it travels to the cell and then back to liver. Okay. When these chylomicron and VLDL, they lose their content, mostly in the form of triacylglyceride or TAG, they will become intermediate density lipoprotein or IDL. And then further losses of TAG within IDL will make it to become LDL, or low density lipoprotein, where it only has cholesterol in it. It is having so much of cholesterol, it's been referred as the bad cholesterol, because a patient with high cholesterol in their body seem to have a lot of LDL. Then on the other side, there's also HDL, high density lipoprotein. High density lipoprotein has no cholesterol or less cholesterol, but it has a potential to take up cholesterol. So this is the good cholesterol, because it tends to pick up all those cholesterol dropped by LDL and bring it back to the liver. So these are all the types of lipoprotein that is found in our body. Next, we move our attention to the contribution of lipid to the cell membrane. First, we look at what is cell membrane. Basically, the cell membrane is made of lipid bilayer, and as this slide referred, the outer plasma membrane will cover the whole perimeter of the cell, and then you have the internal membrane that cover the organelles. Next, we're going to look into the lipid contribution in making into lipid bilayer, and then this lipid bilayer will become the cell membrane, complex structure. And in terms of the lipids or the lipid bilayer, these are the four characteristics that we're going to look into. That it gives rise to selective permeability. It doesn't allow everything to go in the lipid bilayer. In fact, anything got charges or anything that is water soluble cannot go into the cell membrane because of the lipid bilayer. And then you have membrane fluidity, where it actually is dynamic. It's not staying stationary, the lipid bilayer. The lipid bilayer also, if you can cut it at any part of it or any region, it can actually come back to becoming normal again. And then the lipid bilayer, the two layers are not really having the same number of lipid. It is different on one side compared to the other. That's why you are having it as asymmetry. So now we look at the selective permeability. As I referred to you the slide before this, it's the hydrophobic region of the lipid bilayer where it has the hydrocarbon chain, and that part, the head and tail, the tail is the one that does not allow all these ionic or polar substances to pass through. And therefore, to allow these substances to still go through, you need membrane protein or membrane channels. And that's where you have the active transport as well as passive transport, which we will see later. So this slide basically talks about the compartmentalization. Com that means it's having compartments. Here on the left, you can see there is a cell membrane. Okay? And because of the cell membrane partitioning, you can see only substances of red and green, and it doesn't mix with the outside compartment of having blue as well as gray. So this is one way to keep certain substances away from inside the cell. But in actual sense, the one on the right is what's found in the cell, where there is organelles. This could be said as organelles, say mitochondria. And then you can see they are orange as well as yellow substances can be inside the mitochondria, while the green and red in the outer side, which could be referred as cytoplasma. So you can have certain pathways happening in the mitochondria, like citric acid cycle, while in the cytoplasma, some other pathways like glycolysis taking place. So it is separated. Therefore, it cannot have something referred as futile cycle or wasteful process taking place. So it is nicely separated from one place, one area or location for the pathway to take place, and the other one will be inside. So here you have the cell and also what's inside the cell, the organelles. Uh, just want to highlight to you, when we learn about metabolic pathways in biochemistry, you need to know where is the location of the metabolic pathway. So some pathways could be happening in the mitochondria, some could be happening in the peroxisome, also some could be just happening in the cytoplasma. So it's just to know the location of this individual metabolic pathway. So we revisit the fact about amphipeadic, because now we're going to see the contribution of the amphipeadic phospholipid or other compounds that have this amphipeadic structure. The amphipeadic structure is basically, having the uh, charge group, or they call it the head, and then you have the uh, tail, tail being do not like water, while the head, because it has charges or it has a lot of OH group, then it likes water. So head and tail. So here given some or all the amphipeadic molecules there are, you have the phospholipid on your left here. So the example given is phosphatidylserine. So it has positive charge as well as negative charge. So and also on the phosphate. And then it has the tail being hydrocarbon chain and does not like water, so it keeps the water away from it. You also have cholesterol having the water liking OH, and the tail section do not like water. Similarly, you have the glycolipid, the galactose having a lot of OH group, therefore it likes water, and the tail that does not like water. So here given a comparison of two different substances, one that likes water and the other one does not like water at all. The one that is does not like water at all is the triacyl glycerol, because it's made up of three fatty acid. There's nothing that having charges or OH group. Therefore, this one, when you put with water, it stays entirely away from water. Then you have the MP-perdic, that is phosphatidylethanol Uh, it also has its limitation in liking water, because this head group likes water, but this one goes far away from the water. So because of the MP-perdic capabilities, wherever you put the phospholipid, for instance, the head will always be close to the water, and the tail section, as you can see, facing furthest away from the water. So you put water in the middle, what you get is the head, so you get something like a micelle structure. And then if you put water on the outer section, this will be the structure you obtain, where the hydrocarbon chain will be internally inside and the head will be outside. You also have the structure that you find in the cell, where cells will have water environment in the outside and also internally. So this is where you get the lipid bilayer structure, as you can see here. The lipid bilayer structure is the basis in building the cell membrane. So here you see the lipid bilayer, where it can allow certain substances of hydrophobic nature to pass through without any problem, because the hydrocarbon chain of the lipid bilayer allows it. But if it is a charged molecule or hydrophilic molecule, and therefore it will be restrained from passing through the lipid bilayer. So that's where the protein channels come in to help to move the molecules from one part of the cell membrane to the other across. So now we look at the whole cell membrane, not only the lipid bilayer, also other components that come together to make the cell membrane. So you have the cytoplasma to be water environment, extracellular fluid also water environment, therefore the lipid bilayer will form. You have the filament that actually communicates from inside the cell to the outside of the cell, phospholipids. You also have cholesterol. Cholesterol help in terms of the fluidity of the lipid bilayer, which we will see in a while in another slide. You have the transmembrane protein, not only the protein going through being transmembrane, also peripheral protein inside the cell. You have conjugated protein, glycoprotein, with the protein to be buried inside the cell membrane. You also have glycolipid, with the lipid part buried in the lipid bilayer and the carbohydrate to the outside of the cell. Next characteristic that we are going to look into is the membrane fluidity. In terms of membrane fluidity, the lipid bilayer is not static. In fact, it's quite dynamic, where the phospholipid can rotate in its axis, it can laterally diffuse, and it also can do flip-flop, but not much of a freedom when it comes to flip-flop, but more so of rotation and lateral diffusion. And then you can see there that it's been indicated high concentration of unsaturated fatty acid can actually be making the lipid bilayer to be more fluid, because unsaturation will have this bending or kinks, and therefore less space packed together, therefore it can have more fluidity. And then also been stated, when you have more cholesterol to be added, the fluidity is actually reduced, less fluid. So here shown the importance of the membrane fluidity compared to being static. Obviously, having it to be fluid helps in certain aspect of the cell function. So here shown some of it. I'm not going to go into all of it in detail, but one of it is for cell signaling. Just imagine if it is static, then the receptor is waiting to meet its ligand. But if it's moving, if the receptor is moving, it's more chances for it to meet the ligand. So that's the one of the plus points of having membrane fluidity. In this slide, you have quite a lot of information, but the one that is most relevant is going to be on the parameters that decides on the fluidity of the lipid bilayer, hence the whole cell membrane itself. The two parameters that I am referring to is firstly the length of the hydrocarbon chain of the lipid bilayer. The longer the hydrocarbon chain, hence less fluid it will become. On the opposite, if it's shorter, more fluidity the cell membrane will become. Another thing is the degree of saturation. The more unsaturated the hydrocarbon chain, therefore the more fluid the cell membrane will become, because there are spaces between the hydrocarbon chain. So in terms of saturation, it also makes it become less fluid. So this is the effect by the length as well as degree of saturation. So this is just to revisit what we already spoken about. That is, in plants there is more unsaturation. Then in animal fat there is more saturated type of fatty acid. But in terms of the lipid bilayer, this unsaturation plays a role in making it very much fluid in the because of this space here, and also uh, meeting up with the neighboring hydrocarbon chain, the space in between. So what happens to the cell membrane is when you increase the temperature, it will be starting to be fluid and more fluid until it could even rupture the cell membrane, therefore could make the whole cell to die. So just imagine you take a bacteria from moderate temperature, that is 35 degrees Celsius, then you shift it to hot spring. So it become so fluid, the cell membrane of the bacteria, it is possible the cell or the rather the unicell bacteria could die. So what does bacteria do in this kind of situation? What happens is, in this situation, the bacteria plays around with the length of the hydrocarbon chain. When it is put into hot spring kind of temperature, it will cause the length of the fatty acid to increase, and therefore now the fluidity is controlled there in order to not have it to rupture the cell membrane, for instance. So the bacteria plays around with the length of the hydrocarbon chain and also looks into saturation and unsaturation. Obviously, moving it from temperate to hot spring, it will cause more of saturated fatty acid from what was unsaturated. So in this way, it can control the fluidity. Unfortunately, animal cells do not have this ability of controlling saturation, unsaturation, as well as the length of the hydrocarbon chain. What happens is we animals have cholesterol to be inserted in between that space that I just showed you in the previous slide, and therefore it will restrict the movement of the hydrocarbon chain, and hence the whole lipid bilayer will be controlled in terms of the fluidity, and therefore hopefully there is no rupture of the whole cell. So here you can see the cholesterol that has been put in order to restrain the movement of two hydrocarbon chain. So this is how fluidity is controlled with the assistance of cholesterol molecule. So the next characteristics of the lipid bilayer is the self-sealing capability. And this is important because if accidentally there is a mechanical damage or being cut the cell membrane, it's good that the lipid bilayer able to uh, restore itself and therefore back to normal again. So this is the self-sealing capability that is present in the lipid bilayer. It does not rupture for good. And this is the fourth characteristics of lipid bilayer, that is asymmetry. Here it has been found that during the making of the lipid bilayer, there is more phospholipids on one side compared to the other side. And why this is coming about is not really known, as well as what is the importance of having this asymmetry of the lipid bilayer is also unknown. So upon knowing more about the lipid bilayer and how it is formed, and also the four characteristics, now we actually look into another component of cell membrane, that is the proteins, the membrane proteins. In terms of the membrane proteins which are embedded in the lipid bilayer, it could be either integrally embedded or this integral embedment is running across the lipid bilayer, while the peripheral protein will be within the inside cell of the cell membrane of the lipid bilayer. So here, given the contribution of membrane protein, it could be either for recognition, it could be also part of enzyme, the proteins, enzymes, it could be receptor proteins, and also it could be the transport proteins, which can be divided to be passive as well as active. In the slides to come, we get more clarification on these four. Before we look into the protein function, better we learn about this fluid mosaic model. But I'm very certain that you already learned about this fluid mosaic model in your pre-U. You already know that the cell membrane has fluidity. That means it's quite dynamic. The lipid phospholipids are often moving around. But another thing you have to know is it's not all lipid or phospholipid alone. It is also having these proteins, also having glycoproteins, also having glycolipids, and all of them are actually you can imagine as mosaics, the mosaic in the floor, and they are constantly moving around within the cell membrane and never to be static. So they are very much dynamic, and this increases certain abilities, and that's what we're going to discover in the slides to come. So this recognition of protein is vital because the protein, not protein entirely, the glycoprotein, which will have the protein side to be running transverse through the lipid bilayer, while the glyco part is jutting out into outside of the cell membrane, outside of the cell. And this glyco part is, you can imagine it as being like a fingerprint. It sort of like gives out signal whether the cell is self or foreign. If it is doing its job correctly, it will recognize it as self and therefore will not be destroyed by the immune system of the body. But if it's gone haywire, then as in the case of cancer, it might destroy your own cell. Okay, or if a particular cell has been infected and infection is beyond recovery, therefore it will be destroyed by autolysis. So that's recognition of protein. So how the glycoprotein helps in knowing whether it's a self or foreign cell. Then you have the receptor proteins, whereby it is useful for signal transduction. So you have, for instance, insulin. Insulin is a hormone that is released by the pancreas. So then it goes to the cell, say muscle cell, and there is special receptor waiting for it, and the receptor will bind with the hormone and causes a triggering of signal, and therefore it will cause the channel to open up, that is meant for taking up glucose from the bloodstream to inside of the cell. So that's an example I can give in terms of receptor protein. So cells, at the outside of it, there is a lot of function relevant for the usefulness of the cell itself. So here shown there in terms of the binding of the hormone, for example, or messenger or other messengers, which is very specific. It only can accept a particular messenger with a particular receptor, just like lock and key for enzyme. And once it has received its messenger, the signal transduction will take place until it will trigger an activation of another molecule outside or rather inside of the cell, for instance G protein, and this say activated G protein will then go and open up or activate the protein channel to open up, and therefore can then do its function. So in terms of the protein channel, they are divided into two types. One is the passive transport protein that has no requirement of energy in the form of ATP to be hydrolyzed. Then you have the active transport protein, normally from low gradient to more concentrated gradient of molecule or molecules. So in that event, it's going against the gradient, therefore need to use energy, and the energy is normally by ATP hydrolysis. So here given on the left, passive transport through the protein channel that is found in the cell membrane, that is highly concentrated, diffusing to low concentrated area. Then you have the on the right, active transport, which requires ATP hydrolysis, and this energy will be then utilized to drive the low concentrated molecule to an higher concentrated area. So basically, plasma membrane does not only have lipid bilayer, it has also other contributing members, and apart from channels, you also have these glycoproteins and glycolipids, which are conjugated types of macromolecule, that is carbohydrate and protein, and carbohydrate with lipid. And normally the protein and lipid will be in the lipid bilayer, while the other conjugate, the carbohydrate, will be on the outside at the extracellular site. So this figure gives you an idea that it's not only about lipid bilayer, but it has other components or other members also playing a role in making the complexity of the cell membrane itself. So you can see outside of the cell membrane, outside of the cell, there's a lot of activity. You have the glycoprotein, the glycolipid. You also can see the proteoglycan that you learn in the carbohydrate lecture. I just want to show you. So this is the the glyco proteoglycan that you have learned in the lecture for carbohydrate. So now, just to highlight the fact that in the extracellular side or extracellular matrix, there are this layer that is being referred as glycocalyx, and it encompasses of transmembrane glycoprotein, absorbed glycoprotein. You also have transmembrane proteoglycan, glycolipid. So all these must have a function. This function is contributed as a whole, known as cell coat. And this cell coat, as you can read, it protects the cell surface from mechanical and chemical damages. It's a bit slimy because it absorbs water. So basically, if it's a cell, it is not rough on the surface, but rather slimy, and because of that glycocalyx or cell coat, any bumping around with another cell will not damage the cell itself. So it is kind of like a mechanical protection. So if just now we talked about mechanical protection, here you have more of immunological protection, whereby if say there is an infection at the site as shown there, uh, there suddenly the glycolipid glycoprotein will be put like a flag to flag that area, and this will cause the white blood cell neutrophil to recognize that flagged area, and it will then come in to diffuse into that area where got infection, and help to remove the infection altogether. So that's the importance of the glycoprotein or glycolipids that is found on the outside of the surface of the cell.